Hello, Eastview Scholars, and welcome to this edition of Sharp Shorts, where we're going to talk about the Calvin cycle. When we last left and talked about stuff, we had gone through the light-dependent reactions and their need to produce ATP and NADPH to send to the Calvin cycle, and we went through linear and cyclic electron flow. So our next step in this then is seeing how does the Calvin cycle incorporate this energy and reducing agent and carbon dioxide to produce the sugars that we have. Well, it's very much like the citric acid cycle. You are regenerating carbon-based molecules, and this time it's not oxaloacetate, but it's something called ribulose bisphosphate, which is a 5-carbon sugar with a phosphate on each end, so it has lots of potential energy. And it has 3 phases. You're going to fix the carbon with an enzyme called Rubisco. You're going to reduce that down to a molecule which you should be well aware of because it was the same in glycolysis, glyceraldehyde 3-phosphate. And then you are going to regenerate some of that glyceraldehyde 3-phosphate, G3P, into the ribulose bisphosphate again to produce the RUBP that you need to keep the cycle going. Just know that as we look at the picture, to make one G3P, which is the precursor sugar molecule, again, you don't make glucose directly out of the Calvin cycle. You make that 3-carbon G3P that can then be used to make glucose and other carbohydrates and fatty acids and amino acids and stuff along that line. It takes 3 cycles. And in most books and on the pictures that we're going to see here, we're going to illustrate that you've got 3 from your book. They show 3 of these gray balls as carbon dioxide coming in, but each one is being fixed one at a time. So essentially what you have here is 3 molecules of this 6-carbon. So anyhow, the idea is here's our ribulose bisphosphate. This 5-carbon sugar with a phosphate on each end. Now it says you have 3 of them because we're assuming we went through the cycle 3 times. And it combines with this enzyme Rubisco, which then incorporates the carbon dioxide into this 6-carbon sugar with a lot of high potential, but very unstable. It doesn't last very long. And so once you incorporate a carbon dioxide, it splits in half into 2 3-carbon sugars. And if you think then that went through 3 times, that's why you see it says 6 of these. That just means that 6 total on after 3 cycles. You know, each one would produce 2, you have 6 total. Now, that now is a 3-carbon sugar with a phosphate at one end that's got potential energy, but if I were to add ATP to each one of them, then I would make this 1,3-bisphosphoglycerate high potential 3-carbon intermediate that is just a stone's throw away from being glyceraldehyde 3-phosphate. All I've got to do is now take my the reducing power NADPH and knock this down, removing a phosphate group. And so here's my NADP and my inorganic phosphate and my ADP that go back into now the light reactions to keep the process going. Photosynthesis is what's called an emerging property. It was something that developed upon an endosymbiotic relationship that developed that created the chloroplast. So it was something that developed as the earth and living creatures did. So here was my carbon fixation phase. Here was my reduction phase. And again, the reason we showed this with the molecule numbers essentially tripled was because you need 3 spins to make 1 G3P that kicks out to make glucose and other organic compounds. The other 5 that come through here is what the regeneration phase is. And it's a very complex thing to take these 15 carbons and rearrange them, and using 3 ATP because you want phosphate on each end to get 3 of those 5-carbon sugars. So 5 3-carbon sugars to 3 5-carbon sugars with a phosphate on each end, but it recycles and then the Rubisco can be used. Okay. So that's why it takes 3 cycles to make 1 and 5. And the big thing to notice here, and we knew this because it is an anabolic energonic reaction, it sucks up a lot of energy and stuff. To make 1 molecule of G3P, you use 9 ATP and 6 NADPH for every one. So it is a very energy intensive process, but very important since it feeds the world. Okay, and there's our overview now. We've brought in our light and gone through here, and again, some of these sugars can be stored. You can make amino acids, fatty acids, or the sucrose. Export can go and be glucose. Now, C3 plants are very common, and they're called C3 because they get fixed, and that's that precursor, that 3 carbon. But obviously, in the fight for food and competition, there were other things that developed in different environments and different amount of light and carbon dioxide intensity and everything else. And when stomata need to be open and closed and allow gas to go in and stuff. So you have also what has developed C4 or CAM plants that have found a way to follow the process differently based on their environment. And let's go through why. As those stomata are open, it not only brings in carbon dioxide and releases oxygen and stuff and controls the gas flow, but it's also where the transpiration of water occurs. And so in some environments, like on hot, dry days, plants close their stomata to conserve their water, but that limits their photosynthesis because they're not getting the carbon dioxide in to fix, which allows oxygen to build up from the light-dependent reactions. And C3 plants can go through what is apparently looks very wasteful, what's called photorespiration, which just means that Rubisco also loves oxygen. So as carbon dioxide lowers and oxygen rises up, it'll fix the oxygen and use that and throw it into the cycle. And unfortunately what you get is with a 2-carbon intermediate, it does kick off carbon dioxide, but there is no production of ATP without any sugar or anything being produced. And that is unhealthy for the cell. Why is it around? Well, it's believed that photorespiration was, well, it's an evolutionary relic, as they call it, in the starting up of the Earth, where there was a ton of carbon dioxide and very little oxygen in the atmosphere. Photorespiration wasn't that big a deal. And now seems to be also that it has evolved into a safety catch too in certain things, that plants that don't have that don't do photorespiration see a lot more light damage in intense light. So there is benefits that have been derived from what seems to be an extremely wasteful process. The only problem is it drains a lot of carbon fixed by the Calvin cycle. So it does protect plants in some respect, but it's not. Anyhow, what's the deal here? Well, C4 plants, they have found a way to use a different enzyme other than Rubisco that does not have an affinity for oxygen, and that's PEP carboxylase. So what they've done is they've structurally changed themselves to have an extra layer of tissue. So here's the mesophyll cells, and PEP carboxylase is present there. But the Calvin cycle occurs in these what are called bundle sheath cells, and then kick the sugar into this vascular tissue vein. So it's a protective measure when the stomata has to be closed. You still have your carbon dioxide here. You can still go through the process. And the PEP carboxylase doesn't care about the oxygen, so you won't have that photorespiration problem. And so you can see here carbon dioxide comes in, binds, becomes a 4-carbon oxaloacetate, which becomes malate, which then kicks the carbon dioxide into the bundle sheath area, leaving a pyruvate. Now that can go into the Calvin cycle and produce sugar as normal, leaves a pyruvate. This requires more energy than a normal C3 plant because you need ATP to convert the pyruvate back into PEP. Well, where can I get that extra ATP without going through light reactions and stuff? Cyclic electron flow. So you can produce extra electron by going through cyclic electron flow. But that's their spatial way of protecting themselves from photorespiration in times of hot, dry, when the stomata need to be closed. They found a structural way to fix that, okay, as you can see by the overview here. Now, As carbon dioxide levels have risen during the industrial age, and as people talk about greenhouse gases and everything else, the question is which plant will be affected? Well, in reality, it would be the C3 plants. If you think about it, it's going back to an older time when there was more carbon dioxide. So C3 plants will thrive a little better in this because the oxygen levels, they won't have that issue with oxygen levels with the higher carbon dioxide levels and the photorespiration. C4 plants won't be affected really at all because they fix their carbon in the bundle sheath cells and concentrate it there. Now, the CAM plants, and here's the pineapple, CAM plants are your succulents and stuff. What they have done is they have found a way temporarily to change where their stomata are open at night and they can fix the carbon dioxide into organic acids. And this is what we showed here. So they just can't have those stomata open during the day. Those succulents are going to lose too much water and stuff. So they found a way to, instead of doing a structural change, of doing a temporal change and fixing the carbon dioxide in the acids, and then at night, and then closing the stomata during the day and have the carbon dioxide available then to go through light reactions when the light is pumping out the ATP and everything that the Calvin cycle needs. Okay? So finishing up here is just our review of how photosynthesis occurs. It's an emerging property between these two processes that work together to convert light energy into chemical energy, into food, utilizing water, releasing oxygen, and fixing carbon dioxide. You are now photosynthesis experts. Scholar up, Eastview.